Okay, so our next topic is, we have discussed basic aspect about the cell. Now our next topic is cell membrane. Now it is limiting layer in case of animal cell, or the outermost layer in case of animal cell. Now when you observe cell membrane, it is composed of two things mainly, that is protein and lipid. These are the two main components of cell membrane, and apart from this protein and lipid, we also have glycoprotein, glycolipid and cholesterol in cell membrane. Now, amount of protein and lipid is variable. It depends upon the function performed by that particular cell. For example, most extensively studied cell for cell membrane is RBC. In RBC, we have 52% protein and 40% lipid. Now, to study the structure of membrane, or we can say it was possible to study the structure of membrane because of the construction of electron microscope by two persons, that is Knoll and Ruska. These two persons are responsible for construction of first electron microscope. For that they were awarded Nobel Prize, and that immensely helped in studying the structure of membrane. Now, this lipid component is represented by phosphoglyceride, or we can also call it phospholipid. Now, this phospholipid is amphipathic substance, that is have hydrophilic head, that is water loving head, and hydrophobic tail. Okay, this is lipid component, and it is present in form of bilayer. That means there are two layers of phospholipid. We will study the structure in detail later on. Now, this protein component, or the protein present in membrane, is divided into two different types depending upon ease of extraction, that is how easily you can remove protein from the membrane. If it can be removed easily, then it is known as surface protein or extrinsic protein. Okay, and if it cannot be removed easily, then it is known as integral protein or intrinsic protein. Most of the protein are integral protein. Some of the example of extrinsic protein or surface protein are spectrine and cytochrome C in mitochondria. Okay, so we have discussed basic aspect about the membrane. Now we will discuss structure in detail. As I have said earlier, phospholipid is present in form of bilayer. It has hydrophilic head and hydrophobic tail. Now, in bilayer, this phospholipid is arranged in such a manner that this hydrophobic tail is present towards the inner side. This tail is present towards the inner side and head is present towards the outer side. Purpose of this is to protect hydrophobic tail from aqueous environment of the cell. Okay, that is from the water present in the medium, so we have to protect that tail from that water. That's why this kind of arrangement is there. Okay, so this is lipid bilayer. Now we have protein. There are two types of protein on the basis of ease of extraction. Those which can be removed easily are referred as extrinsic protein or surface protein. Now, protein which is embedded in membrane is known as integral protein or intrinsic protein. Now, then we have glycolipid and glycoprotein. So, suppose this is your glycolipid and this is your glycoprotein. Now, this glycoprotein and glycolipid acts as surface antigen That means different kind of membrane will have different kinds of glycoprotein and glycolipid. Then in membrane we also have cholesterol. Now, this cholesterol is responsible for stability of membrane. Okay. Now, what we are discussing, basically we are discussing the most accepted model of membrane that is fluid mosaic model given by Singer and Nicholson. According to this model, icebergs of protein are floating in ocean of lipid. Icebergs of protein are floating in ocean of lipid. Okay, so according to this model, this membrane is quasi-fluid in nature. This is not the first model of membrane. Before this, unit membrane by Robertson, sandwich model by Daniel Devson was proposed, but those models were not accepted because they were not able to explain how transport across the membrane takes place. But because of the quasi-fluid nature of membrane, transport across the membrane is possible. This protein molecule can move within the overall bilayer like this. Suppose this is protein, this can rotate like this, or this can rotate like this. This can rotate like this can rotate like this. So this ability of protein to move within the overall bilayer is known as fluidity of membrane. This is fluidity of membrane, and because of this fluidity, transport across the membrane is possible. This is most important aspect of this membrane, that is membrane is quasi-fluid in nature, and because of this protein can move within the overall bilayer, and this is fluidity of membrane, and because of this fluidity transport across the membrane is possible, and this model is fluid mosaic model. Now this lipid bilayer can show flip-flop. Flip-flop means this can move like this can move like this. This ability of flip-flop is only with lipid bilayer, not with protein. So lipid bilayer can show flip-flop movement. Okay. Now this is the structure of membrane. Now we will discuss function of membrane. What is the function? As I have already discussed, it is limiting layer of animal cell. Now apart from this membrane perform various function, and the most important function of membrane is transport. Transport is most important function. So diffusion across this membrane is known as osmosis. So those substances which are soluble in lipid bilayer can diffuse across the membrane, okay, and that is known as simple diffusion. Now those substances which are not able to cross the lipid bilayer, they require help of transmembrane proteins or these integral proteins to move across the membrane. What will happen? Suppose there is a substance, this is a molecule, it wants to move across the membrane. Now it will bind with this protein towards this end. Now this molecule of protein will rotate like this because of the fluidity of membrane, and that molecule will deliver towards this side. Now this membrane, this protein facilitates the movement of this molecule across the membrane along the gradient, not against the gradient, from the higher concentration to the lower concentration, and this is known as facilitated diffusion. That is, membrane facilitates this protein facilitates the movement of substance from outside to inside or inside to outside. Now, if this movement is against the gradient, that is from the higher concentration, from the lower concentration to the higher concentration, like this, uphill, okay, from this to this. So if this movement is uphill, then there are various pumps which are present on membrane. Now these pumps are responsible for actively pumping those substance against the gradient across the membrane, and during this process energy is required and that energy is provided by ATP, adenosine triphosphate, and such type of transport is known as active transport. Okay, so transport can be by simple diffusion across the membrane, that is the osmosis, or facilitated diffusion along the gradient with the help of transmembrane protein, or with the help of pump against the gradient with the utilization of metabolic energy, and that is known as active transport. Now apart from this, what are the other functions performed by this membrane? It helps in cell growth. If there is growth in any cell, what will happen? Membrane will expand, and without expansion of membrane, growth is not possible. Second, cell secretion. Cell secretion means within the cell, substances are delivered in packaged form between the cell or within the cell, and that substance is delivered in form of packaged material, and packaging is with the help of membrane, and this process is known as cell secretion. Third, formation of intracellular junctions. In animal or in zoology, in chapter tissues, you will study there are various kinds of junctions, tight junction, gap junction, and that is formed by membrane. Next, cell division. In case of plants, animal cell, this membrane will invaginate. Suppose this is animal cell, so this membrane will show invagination like this, and finally this invagination will meet and will divide cell into two daughter cell. We call it cell furrow method, that you will study in cell cycle and cell division. So it is involved in cell division. Then endocytosis, movement of substance inside the cell or outside the cell. That is of two types. If it is solid substance which is moving, then it is known as phagocytosis, or we also call it cell eating. If it is liquid substance, then it is known as cell drinking or pinocytosis. Okay, so these are certain functions which are performed by cell membrane, and the most important function is transport across the membrane. So now we are through with cell membrane. Next heading is cell wall. Now, cell wall is outer rigid structure in case of bacteria, fungi and plants. Now, in case of bacteria, the cell wall is made up of peptidoglycan. In case of fungi, it is chitin. And in case of plants, in case of algae, it is made up of cellulose, galactans, mannans, calcium carbonate, and in case of higher plants, it is made up of cellulose, hemicellulose, pectin, protein, etc. So these are the different components of cell wall in different organisms. Now this cell wall is responsible for a defined shape. It is responsible for defined shape. It provides protection from mechanical injury. If there is any mechanical injury, it will protect from that mechanical injury. Then it helps in cell to cell interaction. Then it forms boundary for apoplastic transport. Okay, what is apoplastic transport? Suppose this is a plant cell and this is another plant cell. The space between this space is known as apoplast. In case of plant, most of the water transferred through or transported via this apoplast, that we will study in transport in plants. So boundary of this apoplast is formed by cell wall. So these are some function of cell wall. Okay. Now, in plant, cell wall is divided into three different components. That is primary cell wall, secondary cell wall, and third is middle lamella. So these are the three different components of cell wall. Now we will discuss this one by one. Suppose this is a cell. In plant cell, we have a large central vacuole. So this is your vacuole. Now, this is your primary cell wall. This primary cell wall is thin, elastic, cellulosic, so it can expand, and thus it is responsible for cell growth. Okay, so if someone asks you कि growth takes place due to expansion of which wall, it is always primary cell wall, because it is thin and elastic, so expansion of this is possible. Now next, growth of this primary cell wall. Growth of this primary cell wall takes place due to deposition of material from within. Okay, so new materials are deposited from the within. Okay, and this process is known as intersuspension, that is deposition of new material from the within. Now, as cell matures, this primary cell wall is not able to sustain the pressure exerted by the content of cell. So there is development of secondary cell wall, and development of secondary cell wall always takes place inner to the primary cell wall. Okay, so secondary cell wall will always develop inner to the primary cell wall. A very important point. And if someone asks you which wall is closer to vacuole or membrane, so it is always secondary cell wall which is closer to membrane or vacuole. So this is your secondary cell wall, and this is your cell membrane. Now, as cell wall is permeable structure, cell wall is permeable. For large macromolecule it is not permeable, but for small molecule it is permeable. Now, so growth of this secondary cell wall takes place due to accumulation of material from surrounding, and this is known as accretion. We call it accretion. So growth of primary cell wall is due to deposition of material from within, that is intersuspension, and growth of secondary cell wall is due to accumulation of material from surrounding, and it is known as accretion. Now, in some cases, like in xylem trichid, there is development of tertiary cell wall, and it develops as a result of deposition of lignin on secondary cell wall. Generally tertiary cell wall is not a common feature, but in some cases you will observe tertiary cell wall due to deposition of lignin on secondary cell wall. Okay, now next is middle lamella. Now middle lamella is cementing layer between two adjacent plant cell. This is your middle lamella. It is cementing layer between two adjacent plant cell. This is cementing layer. Now, this middle lamella is traversed by a tubular structure. This is a tubular structure known as plasmodesmata. Now, function of this plasmodesmata is to establish cytoplasmic continuity between adjacent cells. That means it connects the cytoplasm of two adjacent plant cells. Now, when you will observe it, what you will observe, within this tubular connection there is another tube-like structure, and this tube-like structure is known as desmotubule. This desmotubule is responsible for actual exchange of material. So through this desmotubule, actual exchange of material takes place. Okay, so now we are through with the structure of cell wall.